This is Group 8 presentation on clonal selection theory based on a review of the clonal selection theory of acquired immunity by Jason Brownlee and the clonal selection theory, what it really is and why modern challenges are misplaced by author M. Silverstein. So there are 4 questions that are to be asked in this presentation, and that is what is clonal selection? Number 2, how many theories are there and what are they? Number 3, example for theory, and number 4, how to explain antibody specific production based on this theory. So first of all, we're going to answer what is clonal selection. So clonal selection is the reaction of lymphocytes producing more antibodies in response to specific antigens invading the body. The usage of the term lymphocytes is due to the fact that B cells and T cells are both capable of clonal selection. It was stated B cell in 1955 as T cell has not been discovered yet. But in this theory, the clonal selection theory is also used to tell us how antibodies are produced. So in the sense of that, B cells is the target cell that we're going to use today. So in total, there are 3 theories that give rise to the clonal selection theory that we know today, namely the side chain theory by Paul Ehrlich, antigen template theory by Linus Pauling, the natural selection theory by Nasjern, David Talmage, and Frank McFarlane Burnett, and the clonal selection theory is the product of all 3 that we have stated earlier today. So the sidechain theory is also known as a constructionist theory. It's established by Paul Ehrlich circa 1900, in which he stated that blood cells make side chain receptors capable to bind to all antigens naturally. So this means that information and capability to detect all antigen is actually innate in host and expressed by antibodies. And why does this theory is rejected is that the antibodies also show response to man-made substances. As we all know, man-made substances are not exposed to cell, as thus the cell should not have any information regarding to the man-made substances. And given that the antibodies can also show response to the man-made substances antigen, means that antibodies are do not have all the required information such that they can produce the receptors to bind to the antigens. As such, that is why the side chain theory is flawed. Then the sidechain theory is not also cannot explain how does the secondary and subsequent immune response to the same antigen occurs. Number 2, we have the antigen template theory by Linus Pauling. This theory is an instructionist theory, and he proposed that antigen directs immune response, in which the antibodies utilize antigen as model for self-modification to produce more effective complement. This theory is rejected as specificity of an antibody cannot be changed after its creation due to the central dogma of molecular biology. We all know that information can flow from DNA to RNA and to amino acid, but the DNA, but the information cannot flow back from amino acid back to RNA. As such, the antibody, which is consisted of multiple amino acids and forming proteins, cannot actually revert back to RNA to change its specificity. So now Nasjern had observed the antigen template theory, and he discovered a few flaws that exist, in which that number 1 is that antigen template theory suggested a one-to-one relationship between antibodies and antigen. But actually there are more antibodies produced than antigen in primary response, indicating a one-to-many relationship. This also correlates to the finding that secondary stimulus induce a more vigorous immune response than primary response, and there are exponential rise in number of circulating antibody after first exposure to antigen. There are also more awake antibodies to antigen produced in response due to repeated exposure, and the fact that antibodies' role ended after neutralizing antigen, and they have a low lifespan, which does not correlate to immunological memory and the continued production of antibodies long after exposure to the antigen. So Nasjern proposed another theory, which is the natural selection theory. So natural selection theory is a selectionist theory, and he suggested that the host has a small pre-existing collection of randomly generated antibodies. So the self-antibodies are neutralized to prevent them from causing harm to hosts through negative selection. As we all know that self-antibodies will bind to self cells, thus causing autoimmune diseases. Antigen will naturally select antibodies with higher affinity to it, and Nasjan conveniently suggested that copying errors during antibody production may result in an improved antigen and antibody fit. So David Halmer has suggested that the sidechain theory has a place in the natural selection theory, and he stated that replicating cells is the main feature of immune response. So immune cells must specialize in producing same antibody, which means that the B cell should once has been in contact with antigen and start to produce antibodies, that B cell will only produce one type of antibody, which shows that they have the same specificity. So Burnett was associated with two theories at first, which is the antigen induces the formation of adaptive enzymes that will form a protein with unique specificity. And number two is that the antigen instructs the formation of an indirect template against which specificity might be imposed during protein formation. So the 1949 theory of Burnett's has caused the question of self-antigen response, and he is burdened by the founding by Peter Madawa study on tissue transplant by showing that allografts are rejected while autografts are not, which shows that there are certain response to self-antigen. And Ray Owen studies on cattle actually shows that self-recognition is learned rather than be programmed. Frank McFarlane Burnett extended sidechain theory by focusing the perspective of clone and self-replicating cellular mechanisms during immune response, giving rise to the clonal selection theory, providing justification to the exponential rise of antibodies after the primary infection and the improved secondary response due to um, self-replicating cellular mechanisms. So Nozzle and Lederberg also supported the one-cell antibody assertion, providing experimental proof by speaking of the position model, which is a rapid somatic mutation of immunoglobulin gene, showing that actually that is how a sporocyte can actually produce more than one type of antibodies just in different affinity. So the clonal selection theory is accepted, and they have a small random which initial repertoire which shows that the antigen has general specific the antibodies have general specificity toward the antigens, and negative selection of self-reactive lymphocytes in early embryo is done to prevent autoimmune diseases. The selection of best matching lymphocyte by antigen means that the antigen will choose the best matching lymphocyte which has the higher affinity to it, and the antigen and antibody complex will migrate to lymph tissue in which the lymphocyte will differentiate and into plasma cells that produce a lot of antibody, memory cells that will retain the memory of the antigen as thus as serve as catalyst for a future immune response and production of small amount of antibody to fight against the antigen. So the copying errors during proliferation modify the specificity of the cell's receptors and their antibody to the trigger antigen, which actually allows for improvement in certain antibodies. The rate of mutation during this proliferation process is high relative to mitosis. So if we want to explain the antibody specific production, first we need to go through the chart right here. So we all know that the lymphocytes will bind to antigen, and the antigen will actually choose the lymphocytes with the highest affinity to it. So it will undergo mitosis and will proliferate and differentiate at the germinal center to form plasma cells and memory cells. We all know that plasma cells are activated B cells that will produce um, antibodies with same specificity to the antigen, which means that they will not produce any other antibodies to other antigens. Although we all know that um, antibodies of different affinities to the antigen might be produced. So the rationale of having the somatic mutation occurs is to actually to improve on the antibodies that's produced. As thus, we all know that the antigens will always be in contact with the best antibodies, which means the antibodies that have the best affinity to it. Thus, this will actually allows for natural selection to have better antibodies to be circulated through the body to curb the next wave of same antigen infecting our body, thus preventing our body from getting sick better and quicker. So that is all for the presentation today. Thank you and have a nice day.